Okay, very good. So thank you everyone for being here. This is our MOHCCN seminar series. As you know, we go around the country. We've got clinicians, we've got scientists, we've got trainees, we've got patients, and today we're having talks by people from Quebec, some from MOH-Q, and and we have a patient co-chairing the session with Véronique, so I will let Véronique do the proper introductions. Anyway, welcome everyone.
Merci beaucoup, André. Bienvenue à tous, puis merci de nous joindre aujourd'hui pour, oui, c'est notre troisième séminaire de 2025. Puis comme André a dit, il est présenté par le Consortium du Marathon de l'Espoir Québec. So, I'll start with just a reminder that, as André mentioned, it was a relatively new platform, so thank you for your patience. Question in the box, the question mark, or you can raise your hand and main question. Take a moment to our network members and our speakers and our attendees located all across the country on lands that are home to diverse nations, and population. And I invite everyone here to recognize those peoples and lands that we're talking about. Finally, I would like to introduce my colleagues. Louise is an member of the network's patient working group. And on top of all of the work that she and the entire working group are doing, she generously volunteered to participate in today's event. So thank you so much, Louise, for joining us. And I'll pass it to you if you'd like to tell us a little bit more about yourself.
Good morning, everyone. I am Louise Bird, and I am from Carlisle, Saskatchewan, Treaty 4 area. And I have the pleasure of being part of the MOHCCCN, or MOHCCN, or the Patient Working Group Network, since its inception. And it's a wonderful, I can't say enough good things about it and all the work it's doing for fellow cancer survivors like myself. I've been a cancer survivor since, well, I'll go back to the beginning. I had a rare type of inflammatory cancer. I was diagnosed in 2003. My diagnosis process took about 6 months, and it was a real roller coaster because I actually had to fire my first general surgeon and beg to have another one join my team because the first one wanted to do surgery before doing anything else. And I did a lot of research because I had a healthcare background in nursing. No fancy nursing, just general people nursing. And I was scared. I knew that if it was a really rare type of cancer and it presented as a fluid-filled cyst. I hadn't nursed a child for 14 years, and they had first had thought that I had mastitis. And so I knew from just the research I did that if they cut into that, I could be dead within 6 weeks to 6 months. So I threw out my patient advocate hat and said, no, we've got to think this through. And so I'm still here today because of it. And this was 2003 when this happened. I had my second cancer and I found the tumor in December of 2004. So that was after I had gone through my chemo, surgery, radiation, and they thought it was the HER2 cancer again, but it wasn't. It was estrogen positive. So I again climbed out of the pit and I'm still here today. So, and I have the pleasure of working with all of you. So I want to thank the researchers for all the work you do, the dedication, and the because without use, we might not be cancer survivors. I have the pleasure today, and I apologize in advance because I am going to butcher these names. I have the pleasure of introducing Dr. Anne. Wonderful ladies.
This is Morag Park speaking, and you didn't butcher the names, so thank you. Thanks so much. So I'm Morag Park. I think you'll go into slide sharing mode, Louisa. Thanks. I'm this is really a joint venture with Anne-Marie Mes-Masson, and I am at the Goodman Cancer Institute at McGill University. I just stepped down as director, so and to invest even more in the Marathon of Hope. Go to the next, please, Louisa. What's special about the Marathon of Hope Quebec is that we engaged with the Marathon of Hope initially as a consortium. We recognized the importance of bringing the Montreal together. This is the CR-CHUM, the MOH-C Research Institute, the Jewish General, and Hôpital Maison of Rosemont, together who have over 22,000 new cancer patients every year. And this was together with myself at the Rosalind and Morris Goodman Cancer Institute, as well as the McGill Institute of Genomic Medicine, who was building the pipeline. Bringing a consortium together really gave us both the numbers, but also the ability to bring in new centers to build this consortium. And we've added new centers at Université de Sherbrooke, the Centre de Recherche, Université de Cardiologie et Pneumologie de Quebec à Laval, et the CHU de Quebec at Université Laval. So we're covering much of the Quebec province now and working together. And we have the next slide. One of our original visions, particularly in building the pilot of this project and building the pilot of this consortium, was that in addition to the importance of cancer genomics, to understand the genomic landscape of cancers, we felt it was important to build what we would call sort of companion type technologies to analyze the cancers. And we invested in building what we call patient-derived xenografts, so live tumors whereby we can actually test new therapeutic interventions. We recognize the importance of tumor antigen profiling the immune landscape, particularly at scale, and we use imagery and building microfluidics for high throughput, and more recently adding on really looking at metabolic changes both in the serum as well as in the tumor landscape, trying to identify Achilles heels in different milestones. I think it's important to point out that we started pretty much at zero from the perspective of having a pipeline. One of the strengths in Quebec was the fact that we had very well designated and defined tissue banks that had clinical information as well as high quality tissue. So these tissue banks allowed us to enter into the Marathon of Hope really robustly. But what I think we must say is that in the last 4 years, we've set up a pipeline. And I have to confess, I'm stunned at where we've reached today. So we started our pilot in 2018. Our designation, as with everybody else, was in 2021. We got our first ethics approval by December 22, we had 500 cases submitted, and this is really the start of beginning to ramp up. We got our next REB approval to share data within Quebec in 2023. And by March 2024, we had 50. 1 year later, 2,250 cases, but it really showed that we will scale up and getting that pipeline moving. We finally got the REB approval for data sharing across Canada, which is really crucial to share all the data. And by September 2025, we are now at 4,200 cases that are submitted. Some of these are still in the pipeline, but they're at the Genome Center. So I think that in the last really 4 years, we've achieved an enormous amount. The other major milestone this year was in we deposited our data from the first 3 years of this initiative into the CanDig node. So that's the clinical data, and the sequencing data is all going into that node now. And that part to be shared, obviously, across Canada. Could I have the next one? If we just look at we're 3,000 cases completed. We've got 1,500, as I said, that are still in the pipeline. We have a large number of cohorts. We've got 60 plus cohorts. That really reflects the type of tissue banking that was going on and the depth and interest in building research-based cohorts in the Quebec landscape. We've got over 80 principal investigators, and a number of them, and to be honest, I don't have the number, have been awarded Marathon of Hope Quebec Clinician Researcher Awards. And perhaps Louise will chime in at the end on that number. And we have 200 individuals involved both in the management of this project, but also in the daily research and the bioinformatics. The next one. So this just gives you an overview of the depth. What you will see is that we have multiple disease indications that contribute to the cohorts. We are building multiple translational studies, and we've got both retrospective and prospective cases. And also, we put a focus on rare cancers, and you will hear in one of the presentations about rare breast cancer, but we recognize the challenge for rare cancers, and we recognize that these are really poorly understood, and that the Marathon of Hope was an opportunity to bring these to the table and engage across the country. So the current position, but has really taken on a lot of the leadership in the tissue banking and data management in Quebec. Anne-Marie?
Thank you, Morag. So she said, I'm a professor at Université of Montréal. I am the interim director of the Centre de recherche du Centre hospitalier de l'Université de Montréal for another 12 days, after which I hand off the leadership. So I too will have more time to concentrate on MOH and the initiative. I wanted to just talk briefly about the data portion of this initiative because it's so important. It's not just the biobank samples that we have that makes us rich, but also how we've managed to streamline acquiring and sharing the data that we've been collecting. So one of the things we did is, as a network, we have been working but also how to collect our data. And we use the same software, biobanking software in all of the sites, which basically covers all aspects of biobanking, not only looking at the participant, but having clinical annotation around that participant, but also all their identifiers and the consents that we have to do. We have a very broad and comprehensive consent, which allows the biobanks to rapidly join the MOH and share the data and samples. We have information on studies, but we also have, of course, we track everything that's in our collections, how we process samples, how we allocate them, how we store them, how we share them with other research projects. And those who are interested, what we did for this particular project was to create an MOH by the initiative, we've included it into ATOM, so those using it will have data that's compliant to the standards that were set by the MOH. ATOM installation, so sites we are where we have two banks participating, so at the CHUM, the MOHC, the JGH, and the CHUT Quebec. We had ATOM installation, and soon the MOH ATOM installation that will allow them to participate. Also shared it outside the province, and those who are interested, there's the hospitalier Université staff that has it. I share this with others that would like to have a tool in order to participate in the go about streamlining data, and what's especially important for the Marathon of Hope is continuously updating our clinical data. So we had to find a way to be able to plug in directly into our data lakes. So in Quebec, the tumor registry is known as SARDO, and we have qualified medical archivists that enter data into SARDO related to diagnosis, follow-up. That data is deposited regularly in the data lake of the different hospitals, and this data then is captured into and transformed so that it's compliant with the MOH clinical data model. If there is data missing or if it has to be manually transformed, that's done by the biobank staff, and that data is then collected in the MOH-Q ATOM. In addition, everything that relates to this sample preparation and sample data that is required for the MOH is also collected and stored within this ATOM MOH, as well as the sequencing reports we get from the sequencing centers so that we can have that at hand, so we can match all of that data with the participants. That data then is transferred through SD for Health Globus service to the C3G, where the bento ingests the data, combines it with the genomics data, and then through CanDig is able to share it with the rest of Canada. Can I have the next slide? This was a huge endeavor to be able to put this together. So we just wanted to recognize some of the people who have been instrumental in this. We have a great executive committee that meets monthly as needed. Managers and Veronique here at the CHUM, as well as Louisa Izzy, who is over on the McGill side. We have many of our members in Quebec are participants in the MOH-Q working group, the MOH, the ATOM MOH team. We have data wrangling teams at all of our sites that connect the data lakes with the ATOM MOH to allow us to transfer data automatically. We have site coordinators. We have to thank the McGill Genome Center, which has also ramped up with us in this initiative. We've been bombarding them this year with additional samples, our bioinformatics team, all the biobank staff, the research administrators, and of course our partners, including TFRI Health Canada, our patient partners and our funding partners, because as everybody knows, this is a match program. And so we've also have to thank all of our partners who help us match these dollars for the Marathon of Hope. Next slide, please. I wanted to, since we just had the race, I couldn't help but plug our MOH-Q team, which ran in both Montreal and Quebec. We had set ourselves, I think, a stretch goal, which doubled what we had given ourselves as a goal last year of $10,000. And we reached and surpassed that goal. So we're very pleased. And these are just a few pictures of the day that we spent together, not only celebrating among us, but also talking to the public. They recognize us with our purple shirts and telling back to Veronique to introduce us to our speakers.
Merci beaucoup, Anne-Marie et Morag. Yes, so our next speaker is Dr. Antoine Desilets. He is a hematologist oncologist in the Department of Medicine at CHUM, and an assistant professor in the Department of Medicine at the Université Montréal. He was also recently awarded an MOHCCN Clinician Scientist Award, so congratulations, Dr. Desile. And today he'll be telling us about precision oncology at the CHUM and the Guy Lafleur program. So Dr. Desile, off to you.
Thank you, Veronique, for the introduction. So you should see my slide, right? Perfect. So hi, everyone. Bonjour. So as Veronique introduced, the goal of the presentation today is to give you a bit of an update in terms of our precision oncology pipeline here at CR-CHUM, including more specifically the Guy Lafleur project, which has been running over the last 2 years. So this is my COI slide. And here's a plain language summary. So the Guy Lafleur Precision Oncology Program really wants to transition from a one-size-fits-all approach in cancer care to more of a DNA-guided care. Adults in Quebec only have limited access to the next generation sequencing at the moment, and this program really wants to fill the gap, linking SHIB's molecular pathology platform to also the rest of the MOH network. So our objectives are to provide the first adult precision oncology hub in Quebec to recruit patients, collect samples, and then sequence tumors as a second step. We also want to implement a regional molecular tumor board at CHUM to guide targeted therapies, but also clinical trial matching. And the methods really are tissue blood collection through our biobanks, followed by DNA sequencing with the our collaborators from the and also bit of an overview of what's happening in the province of Quebec in terms of NGS. So giving you a sense of my training. So I was trained at McGill University, obtained my MD degree, and then transitioned to hematology and oncology at the University of Montreal, completing my MSc degree, and then transitioned to 3-year research training at Memorial Sloan Kettering Cancer Center. I was part of the Early Drug Development Service, which is the largest phase 1 unit across the US, with 3 to 4 trials running continuously, and most of those being really targeted on NGS biomarker selection. My mentor was mainly Dr. Alex Drillin and Dr. Ho also, and my interest was, first of all, the implementation and development of targeted NGS, but also whole genome sequencing approaches, as well as pharma-sponsored trials. I also completed bioinformatics and clinical genomics certification, and really giving you a sense of the scope of what could be happening in the US at the moment. So across the MSK platform of the early development service, we see that there's many high impact publications over the last decade, and really the rise of what we call molecular targeting trials. So for NF fusions, red fusions, ROS1 fusions, MET mutations, and many additional oncogenic alterations. So giving you a sense of their platform, so not only is it providing MSK institutional targeted NGS platform, but it's also integrating all of the scheduling services at MSK, integrating the data to a visualization platform through the C-bioportal interface, and also integrating patient-specific queries. So with this alterations to match those active clinical trials are recruiting, and even also to align with the radiology reports, also giving them an update about the latest scan results and the option to notify the clinician. So going back to Canada and also here more specifically province of Quebec. So what do we have? Still have refunding for 8 specific tumor sites in the metastatic setting, and this NGS really is grouped through 4 specialized in addition to the McGill University healthcare system at UCPQ in Quebec and also in Sherbrooke. We are using targeted panel approaches. Here at the CHUM, we're using the AmpliSeq focus panel of 52 genes. There's also additional so we have the approaches here in the process, specifically in the context of non-small cell lung cancer. So CHUM at the moment is the highest clinical NGS in the province of Quebec. Again, also with this for the last year and a half, we have sequenced more than 5,500 patients through targeted NGS approaches, and we are integrating prospectively genomic, clinical, and also therapeutic data from this real-world cohort. Giving you a sense of the implications, so more than 58% of patients had some positive DNA alterations as part of the ampli-seq sequencing, and up to 14% of those had inadequate samples provided for NGS. Looking at more of a specific subset, which is the highest volume in terms of tumor sites, so we're currently studying the impact of NGS inadequacy, specifically in the context of lung biopsies. So many of those biopsies will occur through endobronchial ultrasound guided cytology, so only a small DNA input provided recurrent differences in terms of the result of NGS inadequacy between the CHUM and also the referring hospitals. So as you can see, most of the inadequacy in terms of the first tumor biopsy at the CHUM comes from insufficient DNA material and failure at the step of the extraction when compared to the regional hospitals, when we see that many of those will fail in the context of inadequate tumor purity and sometimes also inadequate tumor sample provided. So this can lead to pretty significant delays in the clinical setting, with delays in obtaining NGS results going up to 6 to 12 months, and many of those second biopsies giving rise to potentially actionable what are we doing to optimize access to NGS in Quebec? So also at the moment trying to get technical team in regional centers to optimize the first tissue biopsy and to increase the tumor cellularity, which is then sent at the CHUM for NGS. As a second step, we're also developing liquid biopsy approaches based on circulating tumor DNA, both in the context of minimal disease assessment. And then more specifically, we are optimizing the platform at the CHUM and developing sequencing oncology program. So taking a bit of my lab at the moment in collaboration with Alex Chang, we're trying to develop specific assays in the context of whole genome sequencing. Also able to earlier detection of resistance cancers, and also in the context of localized cancers following curative intent treatment. And MRD detection can help us into potentially anticipating treatment in the context of MRD positivity or in the context of MRD negativity following surgery or any curative intent treatment. We're able to de-escalate treatment approaches and potentially decrease treatment-related toxicities for patients and also financial toxicity. We're also trying to develop, specifically in the neoadjuvant setting for non-small cell lung cancer, the impact of post-operative MRD in order to intensify treatment, so mainly through intensification strategies such as prolonged adjuvant anti-PD-1 treatment, or also intensification with additional clinical trial options. And this is thanks to the collaboration between our thoracic surgery team here at the CHUM with Dr. Moshe Lieberman and also Dr. Jonathan Spicer at MUHC. Building on those results, we're also trying to develop a multi-omic prognostication model, not only including ctDNA as a dynamic biomarker, but also additional tumor-centric biomarkers in the context of non-small cell lung cancer, including TMB based on whole genome sequencing, PD-L1, pathologic response, and also mostly host-centric biomarkers, such as microbiome metastasis, This is also thanks to the help of our Citadel AI platform at CR-CHUM and our collaborators also, both from the CHUM and the McGill healthcare system. Many trials will also be activated in the context of those studies. So I'm leading a phase 1/2 clinical trial, which is an academic study here at the CHUM, which will evaluate the neoadjuvant regimen of dupilumab and cemiplimab cell lung cancer. And then and increased also active surveillance of complete clinical response following neoadjuvant chemo IO regimens. Then the second step of the presentation really is giving you a sense of the updates, specifically here at the CHUM as part of the Guy Lafleur Precision Oncology program. So as many of you know, Guy Lafleur was a pretty legendary here hockey player who in terms of his treatment was, followed at the CHUM for non-small cell lung cancer, and decided in 2021 that he wanted to partner with the CHUM Foundation program to enhance translational research and also clinical trial implementation. So as of 2022, the first funding campaign began, and which then later gave rise to the precision oncology at CR-CHUM. And April 2022, Mr. Clark unfortunately passed away, but his legacy continues at the CHUM and also to implement clinical research opportunities for patients. So there really are two phases as part of the Guy Lafleur program. The first one, which is innovative projects in precision oncology, and the second one being specifically the prospective sequencing opportunities. The first part of the project was really launched at the end of 2024, with 4 PIs receiving precision oncology programs at the session, and also the official announcement of the goal to accumulate more than $10 million in terms of funding for whole genome sequencing strategies. As a second step, in order to launch the phase 2 of the program, so we were able to obtain Genome Quebec grant as of 2025, with support specifically in the context with 3 main objectives. The first one being certification through ISO standards of a province standard, which provides an alternative to the Optinab clinical sequencing platform. First, so cohort of ovarian cancer patients, so a total of 28 patients underwent sequencing, whole transcriptome also analysis. This one being the Dragon with commercial assay in Illumina, and the second one pipeline collaboration for the C3G and also for Citadel for clinical data integration. And the last part of the project really is to integrate all of those genomic and clinical data for patients through a cBioportal visualization interface in order to harmonize data through MOH and also to clinically to perform specific real-time queries for those patient population. The impact really is to develop scalable workflow here at the CHUM, but also for national collaboration with the MOH network. So this is the overview of the Guy Lafleur Precision Oncology program, really starting from patient recruitment and consent and access to clinical samples through our dedicated biobanking system and going through sequencing, bioinformatic pipelines, clinical grade report emission, as well as molecular tumor board discussions and patient matching to either. All of this to drive research and also give rise to more real-world evidence. This is more of an exclusive also example of our first cohort of patients in 28 ovarian cancer samples that underwent whole genome analysis. This is our real-time cBioportal interface, so evaluating the structural variants, copy number alterations, and also all of the mutations in this cohort, and also giving rise to more of a patient's level analysis in terms of the mutational profile, and also in the next year to finalize the clinical grade molecular pathology platform certification here at the CHUM. Also, we're trying to complete the connection between our pipelines at Citadel and the established molecular tumor board at Q2 2026. Cohorts in order to expand sequencing workflow. We have the goal to acquire in So as a conclusion, I want to thank all of our collaborators here at this network in Quebec, and also specifically Dr. Mes-Masson for trust and support over the last year. I also want to thank MOH for the Clinical Scientist Award that we received last month. So all of our lab will be working on all of those liquid biopsy approaches over the next year, and also continuing to contribute to the Guerrera Precision Oncology program. Thank you.
Thank you for all the hard work you're doing, and also for your team. It was a pleasure listening to you speak. You're very passionate about your project, and once again, thank you. I now have the pleasure of welcoming Dr. Helene Kuzny to speak. Dr. Kuzny is a research associate in Dr. Morag Park's lab at McGill University. And I over to Dr. Helene Kuzny.
My slide, and hear me well?
Yes.
Okay. So thank you for the introduction, and thank you for the opportunity to present updates that we have done here in the last years in rare breast cancer. Specifically, I'm going to talk today rare metaplastic breast cancer. So we know that breast cancer is the cancer that presented the highest incidence across in women across the board. And in Canada is not different. We know that 1 in 8 women will develop breast cancer during their time life, according to the Canadian Cancer Society estimate last year. But the breast cancer is not only one single disease. It's a group of very heterogeneous diseases that can markers like estrogen, progesterone, and HER2. And this expression is very important because it guides therapeutics. But there is also a group that do not express any of these markers, and they're therefore called triple-negative breast cancer. And for those patients, the treatment is standard care treatment is chemotherapy, but a great proportion of those patients will not respond well to this treatment. Breast cancer can also be classified based on depends on where it's come. Or carcinoma that are the most frequent subtypes, but breast cancer can also have another range of one common subtypes that can be very indolent and have a good prognosis. But there is a group that breast cancer that can be a very aggressive disease, and a fraction of breast cancer they contribute disproportionately for the breast cancer deaths because of the biology that is very aggressive, and also because a lack of target. What we understand is that there is a need to better understand the molecular landscape of this disease to be able to inform new therapeutic opportunities for these cancers. Oh, here we go. So with that in mind, when the Marathon of Hope project started a few years ago, here in Quebec, we proposed a cohort to evaluate the molecular landscape of a broad common breast cancer subtypes with the intention to identify targets for precision oncology. Over the last years, we sequenced breast cancer subtypes, and for these cases, we already have the clinical and genomic data, the post-end data analysis is still ongoing. I'm showing you here today that what we observed from the preliminary sequencing results is that the breast cancer can be clustered by different expression profile based on the rare breast cancer subtypes, and it was an information that we have. You can observe it here in this heat map with the top 10 highest expressed genes that clusters, and that we're collecting samples from 3 different centers. If you would have a center has their own protocols sometimes to collect samples and extract the samples. But we observe not the case that most of the expression were the rare subtype and only present the third one, the 3 that were corrected afterwards with some bioinformatic tools. Analysis. One thing that we're very interested in cancer data is rare and very aggressive. And even though we are able to have of these cases, considering the hospitals here in so the metaplastic breast cancer, the name metaplastic comes from metaplasia, that means change in form, and it means that a differentiated cell transform in something else. And in the case of breast cancer, it differentiates in something that is not in the breast. When pathologists see the slides, what it seems is cells that phenotypically looks like squamous cells that we would find in skin, form bonds, or cells that form a cartilage. Spindle-like cells, and all the tumor can be composed of this. And we also see is that often metaplastic breast cancer have distinct histopathological cells and more than one subtype inside only one tumor. So what we understood is that to be able to understand this very interesting biology of metaplastic breast cancer, we would need samples. Hope it was a key to be able to collect and to gather the samples, and also allows us through the Pan-Canadian and technology development grants to use the state or technology companion samples that we had already sequenced and through the consortium. So several collaborators across Canada, and we are able to collect so far more than 40 metaplastic breast cancers, 100 that we are using now for validation in paraffin embedded tissue. And I have to stress out that it was very important also this this network to be able to generate. And so far we have generated patient-derived xenographics of 8 models and also organoids that are going to be used after to test the potential targets that we find in the disease. So what do we know about metaplastic breast cancer? It's a very rare disease. It's often classified as a triple-negative breast cancer, meaning that the standard of care treatment is chemotherapy. However, the metaplastic breast cancer are known to be poor responders to standard of care chemotherapy. And the hypothesis on why the cells are so different to be treated is because we believe that the different cells also have different has been developed for breast cancer triple negative, were designed and tested in invasive ductal carcinoma and not necessarily taking consideration the metaplastic component. And just corroborating this, very a paper coming out telling that neoadjuvant chemotherapy is not what we also know is that metaplastic breast cancer component, if I compare, like for example, this more elongated cells, carcinoma cells we find in the breast, that they have very similar genomic compare these two populations. And if metabolic transition state, we still don't know which to evaluate. To be able to inform the best treatment not only for the ductal carcinoma, but also for the other cells that inside the tumors. So to do this, we I'll say that 5 to 10 years in nice spatial transcriptome technology ready for transcriptomics small in a spatial transcriptome fashion way. So for the markers in the distinct metaplastic population using a platform called high division for the 10x. It's going to be performed in collaboration with Dr. Lupian. And after we would validate the specific markers of each population in a set of FFP. One example of one sample and we selected the sample initially because this is the worst among the metaplastic breast cancer. So this is a very lethal disease. The thing about this tumor is that they are very hard to treat, and it has intensive etiology. Able using of one cells. Find inside the area that is the tumor here in yellow. Away the individual. We can also identify some microenvironment composition also. What I show here. But there are several macrophage infiltration what can make immune system. That this we have in the last couple of years that differentiation enzyme the blocks. Here in this heat map color in green, the expression of LOX in Morag. And what we know, this is LOX enzyme. And the collagen type 1 makes cell more stiff. And this stiffness has been related with poor outcome. And indeed, when we look at the 1 we see that this is heavily expressed. This marker. That expression of LOX and stiffness in cancer is and we started about 2 years ago a collaboration with Dr. South Carolina because he did this compound, and what we are doing now, we are testing the LOX inhibitor in combination with chemotherapy to see if you are able to chemosensitize these tumors. This combination, and we are doing this in our preclinical models that we developed. So far, we have 3 models that is pinball that has metaplastic breast cancer. Here, what I would like to say that we are just able to perform this work because we are able to samples through the Marathon of Hope initiative. And this was very important to gather a great number of samples that we could have biologically meaningful there, and also development of preclinical models. These spatial technologies nowadays are helping us a lot because we are able now to identify very small areas and at the single cell populations inside the metaplastic, and is this sort of technology that we need to understand what's the difference between cell and another inside the tumor. We also were able to show that the distinct cells in this tumor, they have a distinct transcription populations, and they are different in terms of transcription level, and we could identify that would be related to that. And I have to stress out that to test the compounds that we found in spatial transcriptomics analysis. I would like to thank you all in Quebec and across Canada, and also the patients that donate their material and the founders that made this work possible. Thank you very much.
Thank you very much, Helen. It's to see, especially how the network is helping to, make this possible for things like rare cancers to work together across the country. So as I said at the beginning, we now have a few minutes for questions, which is great. Please either put your question in the Q&A, which you can access at the top of your screen, or you can raise your hand by pressing the little hand button also at the top of your screen, and we can unmute you and let you ask your question. To start us, I'm going to see if my co-chair Louise has a question that she'd like to ask.
Okay, thank you, Bernie, and thank you to our speakers. My first question is for Dr. Desilets. Who is eligible to participate in the McGill program? And also, is it open to all cancer patients?
Very good question. So for now, we were specifically targeting the ovarian cancer cohort, which was sequenced retrospectively. The second part of the project really is the select prospective cohort of patients with solid tumors, and we're currently deciding on the specific subsets of patients which will be eligible, and growing and based on this experience, we want to expand the sequencing capacity and eventually at term be able to prospectively sequence patients through whole genome sequencing. All patients will be diagnosed and selecting specific populations for 2026.
Thank you, Dr. Desilets. My next question is for Dr. Kuzny. Dr. Kuzny, can you expand on why national collaboration is so important, especially when you're studying?
A biological meaningful of our findings, we needed to have number of samples, especially there are very heterogeneous among the breast cancer. So if you have only one case in one hospital, another case in another, but we cannot extrapolate this for other cases. So we need numbers to be able to make our findings spread it out, and to be able to find the biological meaningful, you need numbers. We need more samples. And I obviously you have a pan-Canadian here that I would like to say that we are often connecting to get samples, to have the sample sequence, the Marathon of Hope. So it's good to ask her to thank you. Our collaborators have been shipping samples for spatial transcriptomics, and also say that if in your biobank and you flag that you have a metaplastic breast cancer, we are welcome to do all these tests on this. And so that's why we need a great number of people to be able to extrapolate our results for all metaplastic breast cancer, not only one single person.
Thank you. And thank you for the research that you are doing and that both of you are doing. It really hits the heart of somebody who has had cancer. Thank you.
Thank you for that, Louise. André.
Yes, thank you very much. The question is for Antoine. So I was a little bit lost when you said you had like 5,000 samples, and then you were talking about which one you did panels and which one you did next generation sequencing, and then you squeezed in their whole genome sequencing. So how many, so primarily, so how many have you done whole genome sequencing on in these 5,000?
Totally. So there really isn't the main difference between the clinical NGS platform, which was done at the CHUM routinely for all patients across the 8 specific tumor sites. This is the clinical NGS, clinical genomic registry, which is more than 5,000 patients. And then this is in parallel to the actual Guy sequencing strategy, which for now has been focused on establishing the pipeline. So it is a smaller subset of patients, 28 patients specifically, that were selected as part of our biobanks and in the context of ovarian cancer. So you have 28 patients that have passed through the whole pipeline. And the next step is to expand as the beginning of 2026. So 28 patients specifically for ovarian cancer through whole genome sequencing and whole transcriptome analysis. And the second aspect was mostly the clinical NGS with targeted panels, which is more than 5,000 patients.
So far, these are retrospective cases or they're prospective cases? And how, if it's prospective, I'm sorry?
Yep, so the Guy Lafleur currently is still establishing the pipeline, so those were sequenced retrospectively, but the second part of the program really is to implement the prospective sequencing of patients. Yep.
And then shorten the time between the biopsies
exactly.
and the availability.
Working on the pipeline to decrease also the turnaround time and also to improve and optimize the clinical actionability of those results.
Right. Okay, good. Thank you.
Thank you.
Thank you. Debbie, I see you have you should be able to unmute yourself to ask your question.
Thank you. Can you hear me?
Yes.
All right. This message is for Dr. Kwasi. Thank you all for the great presentations. I'm part of the patient working group with the Marathon of Hope, and I'm curious into the triple negative and the samples that you've obtained. In that particular type of cancer in the breast, has there been any ability to gain socio-demographic information within those samples to help guide the studies and the research in terms of who might be more predisposed, I guess we could say, to having this type of cancer versus others? Thank you.
Hi, Debbie. We do have this information actually through our biobanks, a lot of demographic information and social demographic information. I didn't integrate it yet into the analysis for our 40 patient cohort and after the 100, but it's a great point. I think that this is something that should be done.
Thank you. Thank you.
Thank you, Debbie. That brings us to the top of the hour. So thank you again so much to all of our speakers. It was a wonderful session and really informative. So we really appreciate you participating today. And thank you for everyone who asked questions. And thank you for everyone who attended. I think I saw a maximum of about 121 attendees. So we had a great turnout today, which is fantastic. So we will see you soon. We'll be advertising our next seminar for October within the next few weeks. So I hope to see you all again. And thanks again for everyone who participated today. Have a great day, everyone.
Thank you. Bye. Thank you to the speakers.